thank you to everyone who is joining us today for this month's NMDC community conversation. So this month we are going to be discussing accessing microbiome research step by step. So we have some great panelists here today, and I'll walk you through what we're going to be doing for the next hour. So we're going to start with a very brief intro on accessibility in multi-omic microbiome science, and then a little bit of an intro on what the NMDC is and how the NMDC is trying to help tackle some of these accessibility issues. Then we're going to have some brief presentations from our panelists, and then we will have a nice Q&A session at the end, and then we'll have a little wrap up. So Marissa Rudolph is the co-host here. She will be manning the shared notes document, which should be in the chat. So feel free to put any notes in there or questions, and she'll be posting any relevant links and more information there as well. If you have any questions during any of the presentations, feel free to put them in the Q&A within the Zoom, and we will have a really nice Q&A discussion about accessibility in multi-omic research, particularly for microbiomes. So just to get a little bit of background about what the NMDC is. So the NMDC has been piloting an integrative data ecosystem. So the National Microbiome Data Collaborative is the NMDC, and our mission is to provide a gateway to fair or findable, accessible, interoperable, and reusable multi-omics microbiome data, leveraging best practices for data curation and processing. So really great metadata standards and processing workflow standards, and making multi-omic data available to researchers. So we want to standardize everything, make it searchable, make the data fair. And then just a little bit about our resources. So we currently offer the NMDC data portal. So feel free to go to this link and explore any of the multi-omic microbiome data that is currently in there. So we have a lot of different studies from DOE projects so far. So it's a really great interactive website where you can search for a lot of these data sets, and you can even work with them yourself. And so this portal has been recently updated too. So we have a lot of nice data in there. So we ask you to explore the data portal whenever you get a chance. And also for accessibility to the NMDC has developed a platform called Nmdc Edge, which offers gold standard best practice bioinformatic workflows for multi-omic data. So it's offered in this GUI, so it's really user-friendly. So people with any amount of bioinformatics experience can run these workflows too. So just a little bit about what NMDC offers. And then a little bit about multi-omic experiments and kind of what we're going to be talking about today in terms of accessibility. So as many of us are familiar with, the multi-omic experimental process starts with you have some sort of idea or some sort of preliminary data. You write your grants, you receive your funding, you perform your experimental design steps, you perform the actual experiments such as sequencing, you get your data, and then you do your data analysis and your publications, but then that loops back around into this cycle. And so we also have the component of educating the PIs and the researchers that enter this ever-present cycle of the grant writing process and the scientific process. So we have that as an important component into the multi-omic experimental cycle. Put the NMDC logo in a couple places where the NMDC is going to, has been or will continue to tackle accessibility at these particular stages. So these are, for example, the NMDC has multi-omic data that is available to all. So fair data that we are offering to anyone that wants to use it. So that can help get these studies off the ground by giving researchers some of this preliminary data or this data to compare their own data to give the bases for some of these grants that people would like to write. We also have educational programs, and I'll talk a little bit about what the NMDC is offering in terms of accessibility in a little bit. And then these little asterisks are also kind of what we're going to be talking about today during this community conversation, some of these steps in the process where we're going to tackle some of those questions about accessibility in these stages of the process. One thing I wanted to point out specifically, so we're going to be discussing sequencing in particular today, so the multi-omics being the genomics and the transcriptomics primarily. There's also the considerations for the other omic types, but the sequencing is what we're going to be focused on mainly today. So sequencing is expensive, as many of us know, and it can be very difficult to do on your own. It can be difficult to purchase a sequencer, purchase all the instrumentation required to perform high quality sequencing. and it can be difficult to coordinate with other groups or get things sequenced. So the question becomes, how can researchers obtain high quality sequencing data, especially if they don't have a lot of these resources? And we kind of have this classic figure that I'm sure a lot of us have seen of the cost of sequencing going down as time goes on. But a lot of these overall reductions in sequencing costs are not always ubiquitous across different researchers and different institutions. So one thing we're going to be tackling today is how to make a lot of these recent trends towards more affordable sequencing more broadly applicable. So making sure that everyone has access to some of these lower cost alternatives. And we're also going to be discussing new technologies today. So just best practice technologies for multi-omics and microbiome research, and then how researchers can potentially access those. And just talking about some of the disparities that I just mentioned of how these decreases in costs are not always attainable for some labs and for some researchers. So just some questions to keep in mind, and some of these we'll be tackling today during the community conversation, but just the current state of personnel in multi-omics and the trend of where we should be going in terms of accessibility. So thinking about who are the PIs to do multi-omic research, who has the expertise and who can perform these experiments right now, and then really thinking about moving forward, who is being trained? Are we training the right people, or are we reaching out enough to the right groups? How are educational materials being distributed? How are some of these collaborations being fostered? And then which grants are available, who can submit, who is being encouraged to submit for certain grants, who gets these large grants to afford more omics, and how do we encourage more researchers to be able to get these larger grants and do these multi-omic studies and make multi-omics and microbiome research truly accessible? So that it's not leading to some of these cycles of who is funded, and making sure there's always new pools of researchers and diverse researchers. And so one of the big things just to tackle with all of accessibility, so everything that we're talking about really should have a tone of, when we're talking about accessibility, we really do mean extensive accessibility, where multi-omic microbiome research should be accessible to everyone, including all genders, ethnicities, educational backgrounds, sexual orientations, institutions, et cetera. So accessibility doesn't just mean, for the people that want to do this research or can currently do it, but to really enable everyone that wants to do it to be able to perform multi-omic experiments. And a little bit about what the NMDC is doing in terms of accessibility. So like I said, our open data and the data portal really promote scientific discovery and the beginning of scientific questions. And this lowers the barrier to obtaining high quality multi-omics data by providing people with this open data that they can analyze on their own or analyze against their data, or create models or other scientific questions from that. The NMDC is also providing training materials to educate current and future microbiome researchers on best practices and lowering that barrier to entry through educational materials. And like I had mentioned before, the NMDC offers these open source bioinformatic workflows for researchers with any level of bioinformatics expertise. So it's in that nice user-friendly GUI, doesn't require your own computing resources, which I know can be a big struggle for a lot of researchers, is having that computing power and that expertise for the bioinformatics. So the NMDC is really trying to tackle this accessibility in a lot of the products and the offerings that the NMDC provides. And Marissa, actually, who is the co-host here, she developed a really extensive IDEA plan. So IDEA standing for Inclusion, Diversity, Equity, and Accountability. So she fleshed out this really amazing IDEA plan, and we're plotting different actions to move forward with the NMDC and this IDEA plan. And a little bit more about this. So this is based off of obviously the conclusion that diversity and accessibility lead to more science and better science. So we really want to make sure that the NMDC has sustainable partnerships and is making sure that not only fair data, but accessibility in all aspects of multi-omic research are tackled. So the NMDC is turning our IDEA plan into some of these actions. And contact us if you're interested in learning more about what I talked about or anything else that you hear today. If you want to potentially collaborate, or if you want to shape some future actions about our IDEA plan, and anything else that you would like to be involved with. And I'll put a slide up at the end too for getting in contact with us, but we always love to have as much involvement as possible with the NMDC and love to always hear new ideas. And we have an iterative user-focused design process too. So always looking for feedback from the community. And with that, I will just mention that, as many of you know, this is part of a monthly community conversation series. So once a month, we have a different topic for these community conversations. and it brings together members of the scientific community to have some of these discussions that we're going to have today. So we invite you to sign up for future community conversations and to enjoy this one that we are about to start. So I'll just introduce all of our panelists first, and then we can go through all 4 of their presentations. So we're going to start with Sara Bender. So Sara is a program officer at the Gordon and Betty Moore Foundation, who will be discussing foundation-based funding for multi-omics. And then we have Matt Escobar with C Superb, who will be talking about education-based partnerships, such as the JGI Cures partnership. And then we have Adam Rivers from the USDA, who will be discussing accessibility to microbial sequencing and cost reduction strategies. And lastly, we have Ivan Liaschko, the CEO of Phase Genomics, who will be discussing accessibility in private sequencing and some new really cool sequencing technologies. And with that, we will start with Sara's slides.
Great, thank you, Julia, and good morning, good afternoon to everyone. It's a pleasure to be here. For those of you that I haven't met, I'm Sara Bender. I am an oceanographer broadly trained in using multi-omic data sets to study marine microbes. In my current role at the Gordon and Betty Moore Foundation, I support through grant-making projects focused on the development and application of technologies to generally explore our natural world. I just wanted to start with a brief background on the Moore Foundation. We were founded by Gordon and Betty Moore in 2000 and are a private philanthropic organization based in Palo Alto, California. You can see the photo of our building on this first slide. Our organization is set up in perpetuity with the goal, as shared by our founders, to contribute our resources on a scale that we hope can make a significant positive impact on the world for generations to come. So given that, our 2 largest areas for grant making at the foundation are environmental conservation and science, and I sit within our science activities. On the topic of multi-omics, I have 2 slides I'd like to share with you today that highlight some of our work in this space. One as part of our now closed marine microbiology initiative, and the second as part of our new Symbiosis in Aquatic Systems initiative. Both of these efforts either were or are multi-year, board approved and international in scope. We've been working in the multi-omic space for going on 20 years now, and I wanted to mention that because I'm going to give you a couple examples of projects that we have supported in the past and currently, but that's just a taste. I'll be sure to put projects links in the notes that are being taken, and of course, welcome questions when we have everyone back on that panel. Okay, so given that, next slide, please. Okay, perfect. So as part of the Foundation's 15-year Marine Microbiology Initiative, where we invested $250 million in microbial oceanography activities, we targeted these 3 general goals shown here, aiming to remove barriers for our key science focus areas. These included what microbes were in the ocean and what they were doing. Our initiative started in the early 2000s, and so at that point in time, you may recall, we were really kind of at the beginning of ocean sequencing efforts. Our approach is our second goal, development of experimental model systems to study these microbes. And then goal 3, development of technology, methods, modeling, and theory for those science focus areas. Our approaches were to support single investigator awards, multidisciplinary teams, technology and resources, and then of course, community building. After an initial influx of funding for sequencing, including support for Craig Venter's Global Ocean Sampling Expedition, which was in collaboration with the DOE, we then turned our efforts on generating other omic resources. Some of these, and so this is where just a couple of examples will come in. Some of these include diatomic base, led by Dr. Chris Bowler, to develop a centralized platform for analyzing molecular data sets for the model marine diatom, Phaeodactylum tricornutum, to provide a resource for folks to look at not only sequencing information, but all other data that's available for P. tricornutum. Another effort we have supported is UniUke, which is to integrate multi-omic data to develop a universal taxonomy for protists. This work is ongoing and is currently led by Chris Lane at the University of Rhode Island. And more recently, and perhaps more familiar to you all, is support to Bonnie Hurwitz's team at the University of Arizona for Planet Microbe, which of course is connected with NMDC efforts. We were keen to work with Bonnie in part to take all of this rich data generated through the Marine Microbiology Initiative and ensure that it had a place to live and would be useful. And so that work is also ongoing. Bonnie's team is hard at work thinking about data standardization, including using these FAIR principles. Okay, next slide. All right, thank you. So building on the success of the Marine Microbiology Initiative, in 2018, we in the science program initiated the Symbiosis in Aquatic Systems Initiative. So currently this is board approved for 9 years and for $140 million. We have 2 primary goals as part of this work. The first, to also develop new technologies, resources, and theories in this general area. Goal 2, to generate new knowledge of how symbioses function and influence ecology and evolution. Our approaches are similar to those used as part of the Marine Microbiology Initiative to support investigators, to support the development of new technologies, resources, and theory, to support multidisciplinary teams, and then also that key community building component. Since inception, we have supported several multi-omic efforts. One of them is a collaboration with the Wellcome Sanger Institute led by Mark Blackster, and that is to sequence 1,000 genomes for organisms that live in symbiosis with one another. As part of this project, we have a strong community building component, teaching component, collaboration with the Carpentries, which hopefully we can discuss a little bit later. And of course, data sharing is top of mind with complete genomes being immediately released to the broader public so that everyone has access to them through EBI. Another project that we've supported in the early days of this initiative is the Gnps Database, designed to help scientists identify molecules that mediate biological interactions, including symbiosis. And this work is being led by Bill Gerwick and colleagues at the University of California in San Diego. So we are already seeing evidence of new collaborations and of folks beyond these pilot efforts in the Symbiosis Initiative really collaborating across our grant portfolios. We have investigators with certain data sets now reaching out to, for example, the Wellcome Sanger Institute and trying to work together. We're in the early days of this work, but it's really exciting and promising to see what's been done so far. With that, I will pass the mic. Those are my few slides. And again, I will post links to a lot of these projects that I mentioned and happy to take questions later. Thank you.
Perfect. Thank you, Sarah. So next we're going to move to Adam, where we are going to change the slides. Okay, take it away, Adam.
Thanks. I'm Adam Rivers. I'm a computational biologist with the USDA Agricultural Research Service. Next slide. I'm part of a unit that does research into genomics and develops computational tools for agriculture, or I'm one of 4 PIs in the unit. We have 6 people working on different areas of that and a couple of open positions. I can, next slide, talk a little bit about what we do. So our work falls into the categories of metagenomics of insect vectors, animals, plants, and we develop computational tools for metagenomics. We do work on image and time series machine learning methods. We do work on metabolomics of volatile organic compounds and learning from mass spec signals for volatiles. And we work in engineering microbial consortia, doing statistical modeling and developing optimization methods. Next slide. Last year I had the opportunity to be a co-author on an interesting paper on advancing equity and inclusion in microbiome research. And there's a lot of takeaways from that, but one thing I wanted to talk about today was the importance of costs of doing microbiome science as a barrier to inclusion. In addition to being a barrier to inclusion, it's also an issue because it skews the samples that we get in terms of our large global sample collection towards wealthier countries. And it has even for researchers that have good funding options, it skews the kind of questions that we can ask by limiting the number of samples that we can collect. Next slide. So I don't think that we need to accept the $100 metagenome, the $150 metagenome. I think that there's a lot that can be done to reduce pretty dramatically the costs of the types of microbiome sequencing we do. One way to do that is a method called value engineering. It's a systematic engineering method to reduce the cost of products or services or improve the value to a user by changing the product itself. So part of it's pretty self-explanatory. You look at a process, you find the most expensive parts of it, and you start reducing those costs systematically. The second part's a little less intuitive, and that's you have to ask, what are the features that I currently have? So right now for a metagenome, we have a certain read depth, we have a certain phylogenetic and functional gene resolution, and start to say, well, what if I tweaked what those I guess outputs are of that product? And so doing that can be a bit tricky. If you think about the process of sequencing right now, it's a fragmented process. A certain company sells you the extraction kit, another company does the sequencing. There's nobody kind of end-to-end looking at everything. For an individual researcher, there's a big risk, right? You spent a year collecting your samples in the field. You're not gonna mess with the extraction protocol or the library protocol. It's too risky for you to do that individually and optimize individually. And even if you did, now you have to justify that to the reviewer when you go to publish your work, right? So there's not a lot of incentive as an individual to fix these problems. Next slide. I want to make the point that the price really does fundamentally change what you're getting. So if you have a $5 metagenome versus a more typical $150 metagenome, you have a different product. You're going to ask different questions with a $5 metagenome than you are with an expensive one. You ask more statistical questions rather than descriptive questions. Even fundamental things like, are you going to run a QC gel when you send it to the lab? Probably not. If running a QC gel costs more than the whole sample does. So of course, there's going to be some trade-offs. Obviously, if you're paying $5 for a metagenome, you may have lower phylogenetic resolution or shorter reads or have some other trade-offs. But having the option to have a cheap metagenome or an expensive metagenome that's really deep gives you the ability to design different classes of experiments, which I think is powerful. Next slide. So let's talk about a specific example. For a typical amplicon experiment, you might spend $3 to $4 extracting it. Now you've got all these precious samples. You don't want to just ship them. You want to ship them on dry ice because you want to be as careful as possible that you don't lose any. Next slide. And then, so that part is pretty expensive in and of itself, but then you ship it off, you may be paying $80 to $100 per sample. And that's coming from a lot of different steps in the process. So they're doing QC, even things like normalizing your read light, all the samples to put into a multiplex library can cost a good bit. The degree of multiplexing can drive this. So there's a lot of factors that go into this process. Next slide. I want to talk a little bit about how changing one step in a process can have knock-on effects for cost reduction. So this is a pretty neat technique for doing DNA extractions. Basically, this method, you grind up your plant tissue, stick a piece of paper in the first ground up solution, stick it in a rinse solution, and you're done. You basically can take this paper and you can do PCR directly on it or you can dry that paper and do additional things with it. Now, something like that, it's going to reduce the cost of DNA extraction. But now let's say you've got your DNA on paper. Now it's stable. You don't have to ship it on dry ice. So you've knocked off costs there. Similarly, if you go further down the process, you're able to increase multiplexing and shift from a platform like MiSeq to a multiplex platform like NovaSeq. Maybe you've reduced the cost for a particular point down to where you can reduce quality. You don't have to do the quality control gel. So you start to see these sort of incremental effects as you change a particular component, reduce the cost of a particular component in a process. All right, next slide. So what could it take to make a radically cheaper metagenome or amplicon sample a reality? Well, I think we need to fund value engineering for the whole process, because I'm not aware of a lot of people doing that. I think that if you're not going to sell the end-to-end process as a kit, you need to have really good validated methods that are developed by a trusted organization like this one, or JGI or NIST, because nobody's going to risk their precious samples on something that they found randomly somewhere. If you develop a tangible biological or chemical component, it's potentially possible to have it released by an organization like AdGene, that's a nonprofit organization. There are some great services right now for doing fractional sequencing, like Wisconsin offers 1 million reads for $6.65 plus a library prep charge. Those kinds of fractional services allow us to multiplex better. And finally, if we're able to, through funding, develop a whole process for these different types of amplicon samples that has reduced cost, then we could potentially kind of bundle that technology and not exclusively license it to a wide array of sequencing providers and allow them to improve competition. All right, that's
perfect. Thank you, Adam. And then let me just switch back here. Okay, great. So next we have Ivan Liachko, who is going to talk about some of the work that Phage Genomics is doing.
Thank you guys for having me. So my name's Ivan. I'm a founder of a small company in Seattle called Phage Genomics. We do a lot of various genome sequencing, genome analysis stuff. One of the main foci is in metagenomics that I'll talk to you about. And so what I'll do is I didn't want to make this like an ad for the company, but I'll just mention sort of what our technology is, but I want you guys to think sort of vis-a-vis the topic of this conversation is really what we're trying to do is we're trying to essentially simplify the whole metagenome assembly and metagenome sort of discovery process. So that flow chart, that pathway that you saw in one of the earlier slides, we're basically trying to shrink a lot of that because I think a lot of the cost and a lot of the barrier to people getting into metagenomics is not just financial costs for reagents and things like that, but there's also your time, right? And so if you can go to the next slide, what I'll do is I'll talk a little bit about sort of what we do and what our technology does, and then I'll mention towards the end of sort of how we're leveraging this to make your lives easier. So essentially what we're doing is solving what I think is probably the biggest challenge in metagenomics as sort of a science, and that is that when you're sequencing and a bunch of unknown unknowns, you then have to figure out sort of where do all the DNA pieces go? Where do they belong? So if you sequence lots of unknown organisms, you have to figure out who's there, sort of what's their genomic content. If you're ever dealing with mobile elements, if you're looking at viruses, you're trying to figure out which viruses infect which hosts, which bacteria, which microbes. You just don't know that because once you've sequenced purified DNA, right, the phages have all fled the scene. They're no longer connected to their hosts. Plasmids bearing antibiotic resistance have all separated from their hosts, and so you can't really it's very difficult to reconstruct genomes, reconstruct phage host connections, etc. Sometimes we joke that our motto is turning metagenomics into genomics. And so let me show you what it means. So in the next slide, this is what our technology is. So we have this technology that was originally developed for looking at genome three-dimensional folding. It's called proximity-guided metagenomics. So essentially, the way it works is you start by taking a microbiome sample of some sort of mixed community. Maybe it's a piece of soil, maybe it's some poop, and you treat it with formaldehyde to cross-link. And what happens is the formaldehyde goes inside the cells. It creates physical junctions between DNA sequences that are touching each other in 3 dimensions, right? All the genomes are folded up like little hairballs. And you create junctions between sequences that are touching each other inside those hairballs. And then, long story short, you extract them. They're kind of like those little glowing red spots in the left figure. You extract them and you sequence the junctions. And then what you can do is you can take your data and put it together with sort of regular shotgun sequencing. It allows you to say which sequences started out inside the same cell because they were touching each other before the cells were broken. So if you go to the next slide, beyond some formatting funk, basically those black lines are all supposed to be little arrows. You can essentially say, okay, in my metagenome, these sequences were all touching each other, and therefore they belong in one organism. These sequences were all touching each other; they belong in another organism. If you have a virus floating somewhere in your metagenome, you can say this virus was touching this genome because that was the cell that it was inside before the cells were broken. Same thing with plasmids. And so essentially, what we do is we deconvolve metagenomes, right? So our little motto in the very first slide was blueprints for biology. The whole point is what we do as a company is we make genomes, we reconstruct genomes. And so we do that for metagenomics. And so next slide. And so we, kind of the typical way people look at metagenomic data is through taxonomy different driven approaches, whether it's 16S or low pass shotgun sequencing, etc. Everything we do is around some sort of assembly. So we assemble genomes, we assemble metagenomes, and that's sort of what you're seeing is data on the left and on the right from the same set of samples. This is from a fecal microbiota transplant where D stands for donor and B stands for baseline or the patient. So you can see like you have a donor with a big rich microbiome, you have a baseline sample, a patient's been sitting on antibiotics for a long time, and then you do a transplant. And you can sort of, we enable genome resolved metagenomics by assembling all these things. And now you have reference libraries of novel genomes that nobody's ever seen before. You can see what they're doing, what their gene content is, etc. And we go one step further. If you go to the next slide, remember, blurry, cool. There'll be a CRISPR one later, but essentially beyond just kind of assembling genomes, we can tell you, we can see in the data which phages are coming out of which hosts and which antibiotic resistance genes are coming out of which hosts. So on the left, you have AMR genes, antibiotic resistance genes, and sort of which bacteria they're in. And then we can also, so we can sort of tell the antibiotic resistance repertoire in all the different mags. And then we can tell if it's being transmitted by a plasmid, which is the yellow ones, or if it's integrated in the genome, those are the blue ones. And then on the right panel, those are your viruses, those are the phages. So we can simultaneously track the movement of mobile elements like plasmids and AMR genes, and also figure out where the phages are coming from. This is all done out of raw samples. There's no culturing, just this funky shotgun sequencing trick. So if you go to the next slide, please. This doesn't have to stop at humans. So we do this on everything, right? So we operate, we're a company, right? So we work with customers. Most of our customers in this space are researchers. And so, what they buy our kits, they send us samples for service processing. And in the end, you get these sort of data sets. So we get everything. We get water, we get coral reefs, we get human poop, we get poop from different animals, we get poop from celebrity animals. Of course, lots of sewage and wastewater. So pretty much anything, underground caves, and as you can see, there's just there's so much to be discovered in these samples. And so our goal has always been to try to make it as easy as possible, because we forget that even if you have the data, you then have to do something with it. You have to figure out well, how do I make sense of all this insane amounts of data? So if you go to the next slide, so how do we simplify access to metagenomic data? Again, we're doing this as a company, right? So you can just click through to the end of the slide. There's going to be a bunch of little pop-ups. So we've developed a bunch of, we have a special preservative for samples that lets you keep samples intact for a long time. We have kits that we sell to people so you can generate your own data, but we will also do it all for you. One of the things that we find is that trying to control cost by buying kits is definitely a thing. And in the metagenomic space, that's of course a big problem when you're trying to process lots of samples. But we also have lots of people who just don't want to do it themselves. And the way that they get accessibility is they're just like, can you just do all of this for me? And then I will go and publish my paper. That's one way to shortcut that loop. And so we build the kits, and also with all our kits and all our services, we have an integrated analytic platform where basically whether we generate the data for you or you generate it yourself, you can just upload it. and all that analysis happens automatically. And so in fact, the analytics are included with all our kits. And so we sort of, I've been an academic for 25 years before funding phase. And so I know very well kind of like the stress and the time it takes to do all this stuff and try to learn how to run all the different pipelines. And so one of our missions is to bunch it all together into one combined sort of solution, so that you have a sample, you can send it in, and you don't have to worry about learning how to run 7 different tools. And so it's all automated. You don't have to do anything. We just kind of give you the results, and then you can go and publish your papers, and we will help with that. And next slide, please. And so the summary of this mini talk is, there's this new technology for new sort of deep dive microbiome discovery with viruses, with AMR, etc. We sell kits and services to make it easier for people. I know that it's not going to be cheaper than doing it yourself, but it's going to be cheaper than trying to do it yourself and failing at it. We can short circuit a lot of that work. Next click, please. Sequencing services have been mentioned. One of the advantages of being a commercial provider is that we have access to the big machines. So we do so much sequencing that we cram everything into the highest throughput machines possible. And in fact, even the cheapest numbers that the previous speaker showed, we actually charge a lot less than that. We charge probably about 20% less than that. And it's because we can take advantage of scale. Like if you're sequencing things in the MySeq, you're pouring so much money down those machines. And of course, we also have a more sort of regular track record, right? So the other problem with sequencing is the queue. How long do you wait to get your samples done? And we have a much more sort of regular process because we have this pipeline of samples coming through. And then next, and we have these automated analytics that are included, again, to make it easier for you to just kind of like, I have my sample and I have my data, without a super complicated process. Next slide, please. And the other thing that we do, and I think we're a little bit different than most, I'm not saying we're super unique in this, but we're super engaged with academics because our technology is sort of new and weird and it does something you can't normally do. We actually help people publish quite a bit. So, and if you can go look, we've published, I think, like well over 100 papers with our customers. We help you promote papers, right? We have Twitter, we have marketing, we do all the stuff. So when your papers come out, we bump them up, we write blog posts, etc. And so we're a company, this is not pretty straightforward about that, but we also engage with you because we're kind of like a very heavily involved in the academic community as well. And so I think that's all I have for this talk. I think that's my last slide.
Fantastic. Thank you, Ivan. And then lastly, we have Matt. And take it away, Matt. Matt, are you on? I see the video going crazy again.
Can you hear me now?
Yes.
Okay, I'm going to turn the video off. Lesson learned, do not get a new computer refresh the morning when you have to talk at a little session here. So hopefully my computer stays functional through this. My name is Matt Escobar. I'm a faculty member in the Department of Biological Sciences at California State University, San Marcos. I actually don't have extensive background in microbiology, microbiome research. My training is in plant molecular biology. But over the last few years, I've gotten sort of peripherally involved in microbiome research and through a collaboration with the Joint Genome Institute. We're trying to bring sort of microbiome research to maybe a community that maybe doesn't see it very much, and that is undergraduates. And we're doing that through course-based undergraduate research experiences for cures. So next slide. So I'm down here in San Diego County. CSU San Marcos is one of 23 campuses of the California State University system. We're the largest university system in the US. We're primarily undergraduate institutions, which differentiates us from our sister campuses, the UC University of California campuses. temperature focused, more on graduate education research. Collectively, our 23 campuses have an enrollment of roughly 500,000 students. And those students are about 50% underrepresented minority, about 50% low income, and about 40% of them are first in their family to attend college. So a very diverse group of California, primarily California residents. Next slide. So I suspect that everyone in the Zoom meeting was trained in research using the apprentice model, which is basically the idea that one enters a faculty supervisor's lab. One is trained by potentially the supervisor, but more likely a more experienced graduate student or a postdoc or a technician in the lab. And that over time, given the mentorship of those folks, one gains experience and expertise in research. That is absolutely the gold standard, but it also is a very low capacity model for research training. Just to give an example, in my research lab, I have done extensive training of 39 students in 16 years. And that's great, and those students have gone on to do many great things. However, it's obviously not particularly scalable to large numbers of students. You're not going to push 500,000 students through that model. A course-based undergraduate research experience, or CURE, does have that higher capacity. The idea here is you integrate some type of research into a course that students are already taking. It's not going to be the same level or intensity of an apprentice model, but it gives everyone a taste of scientific research. And I would say, in general, genomics research is very amenable to adaptation to its CURE. Next slide. So the CURE that I and some colleagues have developed over the past 5 years started with this paper, Metagenomic Discovery of Biomass Degrading Genes and Genomes from Calorimet. This was published in 2011 by Matthias Hess and colleagues at the Joint Genome Institute. Matthias is pictured in the upper left corner there. And this was at the time the sort of deepest dive into the Calorimet metagenome. And the goal here was really to identify enzymes which could potentially make cellulosic biofuel production more efficient. So identifying novel carbohydrate active enzymes, which could help in degrading cellulose and potentially be applied to biofuels. In this paper, about 27,000 novel genes that encode potential carbohydrate active enzymes were identified, and 90 of those were actually tested. So 26,000 some odd potential carbohydrate active enzymes were not further investigated. And so that was really the starting point of what we do in the classroom with undergraduates. So next slide. So I teach a course called Molecular Biotechnology. It's a required class for all of our biotechnology majors here at Cal State San Marcos. I obviously won't have time to go through all of this, but the bottom line is we take the metagenome data that JGI generated. We target particular genes which have not previously been studied from that metagenomic data set. And those genes have glycoside hydrolase domains. And the students will then, starting with Calorimet, either fluid or biomass incubated in Calorimet, that's donated to me by Matthias Hess. The students will isolate DNA, amplify a novel gene, insert it into an expression vector, and express that gene in E. coli with his tag. We then attempt to purify and characterize the corresponding protein and determine whether or not it has the ability to break down any target carbohydrates. And these steps are done typically with students who, maybe have picked up a pipettement once or twice in their lives. We do this over a 15-week period. Each week we have 1 3-hour lab. All right, next slide. So some students actually go on and take this further beyond what we're able to do in the class. So this is a poster that was presented by 2 undergraduates who took the class in fall 2020. I think we were one of only 2 classes that were still doing face-to-face labs at that time on campus. But these 2 students, Madison Stewart and Cheyenne Black, continued research in this realm in my research lab, and then later on presented their findings at the Joint Genome Institute Genomics of Energy and Environment meeting in 2021. Next slide. So while all students don't get hooked by research, they may not go further. What is clear is that almost all of them understand that they're involved in a research project and they're creating new scientific knowledge, and that the dark blue bar there, and that the findings of their research project were giving them a sense of personal achievement. That is, they're getting, again, a taste for research, they're understanding what it is, and that's linking them or sort of helping to establish identities as scientists. And again, for many of the students in the CSU, they're not coming from backgrounds where you know, they would just automatically step into a classroom and think, oh yes, I'm a scientist, I can identify with this. Next slide. So while Matthias Hess and I originally developed this Cure curriculum for my course in San Marcos, I've subsequently shared it with colleagues at 3 other CSUs, at CSU Humboldt, San Jose State University, and CSU Monterey Bay. And so currently it's sort of variations of this microbiome-focused cure are taught at these 4 universities, and about 100 students a year are involved in doing this research to characterize these novel carbohydrate active enzymes. Along the way, we've gotten some funding from JGI that has allowed us to synthesize 100 sort of larger sized genes from the metagenome data set with GH domains. Those were chemically synthesized and inserted into expression vectors, and we're currently working with those. This allows us to skip all the tedious cloning parts for some of my biochemistry colleagues who just want to get down to the enzymatics. And we're currently working further with the Joint Genome Institute to try to expand this model beyond what was just sort of a spontaneous event that happened between myself and some collaborators at JGI. We're trying to get CSU faculty in general more involved in cures, and we're working toward a process whereby JGI sequencing, DNA synthesis, and metagenomics resources may be available to CSU faculty in the service of creating these sorts of cure experiences for students. And that's all I have.
Perfect. Thank you to all of our panelists for those great presentations. Let me just start my video here. So we have about 10 minutes for a panel discussion. So for any audience members, feel free to put any of your questions into the Q&A. And I will start off with a question for any of the panelists, but how can biologists, geneticists, and other scientists break into the multi-omics sphere? So we can break that maybe into 2 parts of how can people maybe doing one type of omics get into the multi-omics sphere, and then how can any researcher start doing some of this sequencing and multi-omics work? So I'll open it up to any panelists. Okay, Ivan, you look like you want to say something.
I'm trying to figure out if I should raise my hand. No, Sarah raised her hand, and I didn't, so she should go first.
Okay, sorry, did
I'll
not see that. Sarah, go ahead.
Yes, I'll give this a go. Noting, Julia, in your presentation, you provided some great resources for how folks can access this data. So I want to just amplify that because that was some of that was even new to me. I was thinking about this question a little bit and wanted to use this as an opportunity just to mention a couple of other organizations that happen to be grantees of the foundation, but I think are doing really wonderful community building work, and where it would enable someone to access new areas and new methods. One of those is the protocols.io platform, which is open access methods sharing repository. They have been a grantee of ours for many years. It's free to join, and it's a great way to look for protocols and then reach out to the authors of those protocols with questions. We've certainly heard of new collaborations stemming from those conversations on that platform. So wanted to amplify their work, and then just mention kind of other efforts, training efforts. So I'd mentioned the Carpentries, also a grantee of ours. They run foundational coding and data science skill courses to researchers worldwide. I'll put a link to their website in the notes. A grantee from our Marine Microbiology Initiative, Matt Sullivan, has run some great webinars on Biomics. So kind of to hat tip to Ivan's presentation, but methods for analyzing data sets and looking for viruses in there. And those are all recorded, and those are on YouTube. And then through the grant with the Wellcome Sanger Institute, they're also running virtual courses, and they often have in-person courses available too at low to no cost. So there's a lot out there. I think the challenge is just, where to start in one's online digging, but I'll put those links in the notes.
Perfect, thank you. And then Ivan will hand you the question next.
Yeah, no, those are really good. Protocols.io is great. And other resources. So my answer was gonna be a little bit different. My answer was gonna be more along the lines of what does, when you say how does a researcher go about getting into multi-omics? What I hear is really the real question in my space is, how does a wet lab biologist get into omics? How does a wet lab biologist start doing genomics or higher throughput stuff? That's usually the question I see. And I was one of those people. So I did, for most of my academic career, it was all pipettes. And then I learned how to, do genomics and write code and stumbled into that. And I think that's what people struggle with in my sphere quite a bit. And I would recommend, if you're there and you're trying to figure out how to get into genomics or other omics, people will tell you, just play with it, just do some stuff, play around. And that is not helpful advice. What I would say is pick up a specific question you're trying to answer about your research and then figure out how to do it. Don't try to just do genomics for the sake of doing genomics, for example. Say like, I have a strain, it's doing something interesting. I want to figure out why. And I need to sequence it or something. And like, make that little toy example your practice run. And focus on really little bites, because that will give you confidence. Then you can start doing big things, because people just go, oh, I'm going to learn this big thing. I'll get there somehow. I'll play around. That is not helpful. Manageable goals having to do with your current research, and use them as like little stepping stones to learn more.
Yeah, two great answers. I think those are fantastic. And I'm going to ask another question to move this along, and then I think I have some more targeted to Matt and Adam as well. But what kind of partnerships and potentially unconventional partnerships can be formed in order to make multi-omic research more inclusive and increase accessibility through partnerships? And I think I'll pass that one to Matt first, if you don't mind, Matt.
Sure, absolutely. I'm going to turn this. So, I have to say that this partnership that developed between sort of CSU San Marcos and JGI was a completely spontaneous thing that happened because a director of JGI and I happened to be at the same meeting and got to a conversation. And so, that's not a sort of a model that's going to apply broadly. And I think both from the educational side and from kind of the research institute side, we need to do a better job of making connections where those are desired. What I hear a lot from JGI is, and this may be true of you folks as well, is that there's a great deal of interest in reaching sort of a broader community than just the group of current microbiome researchers. And I mean, I can say that both the UC and the CSU has that in spades. So being more intentional about making connections and figuring out where our interests are aligned, I think it's really just a matter of getting those conversations started. But I don't have a great idea about kind of instituting programs per se, but that is really what I think is needed is a more formal outreach.
Fantastic. Thank you, Matt. And I think I can do maybe one more little question, which might include final remarks too. So to Adam, do you have any priorities moving forward in the accessibility space of things that you think are most important to push for in the accessibility realm moving forward, or things that you think all researchers should be considering? And then, yeah, maybe your final thoughts too on accessibility and multi-omic microbiome research would be great.
Sure. Yeah, just because of the charge of our unit, we've been focused a little more on the methodological side, although from a perspective piece, we see the whole all parts as being very important. But we're working on that, doing a little bit of pilot work with some of the cost reduction, starting with amplicon sequencing, because we find that a lot of people getting into metagenomics at USDA, kind of start there where the data is simpler, and we'd like to kind of offer some kind of on-ramp products for them, and then extend that kind of what we learn out to the broader community. So.
Great, thank you. And then I will maybe go around and have one last word from everyone of maybe the most important thing you see for accessibility moving forward, or any wrap-up comments that you might want to include. And I can start with Ivan.
Well, I think the most important thing is people even like thinking about it and expressing interest in it. That's the step one. And I think just deciding that, we'd like to invest in accessibility, or we'd like to open up new doors and things like that. And I think conversations help quite a bit.
Perfect, thank you. And then we'll go to Sara next.
From a foundation and funder perspective, I would just encourage folks on the call that don't hesitate to reach out to funders, ask what folks are working on. If there are projects that you're interested, we can certainly connect you to those PIs. We're not able to support all of the great work that comes across our desk, but much of what we do is also network people in. And the highlight of my job is to get outside of foundation walls and just hear about the work and share it with others. So I'll put a link to an email listserv we use for occasional touch points with our own work, but also please don't hesitate to connect with me through email.
Fantastic, thanks. And then lastly, Matt, any wrap-up comments from you?
So just that I again, reinforcing this concept that the mentorship model of research training and creating research that's accessible is not going to work for everyone, and we need to increase research or access to research and involvement in research among all undergraduate students.
That's great. And I recognize that we are at time, and then I just wanted to share one last slide here. Just about the NMDC to bring it back around to this community conversation, but part of this accessibility is making the NMDC accessible to all of you too. So like I said before, we would love for you to get involved. Here are some of the resources just about the NMDC. If you would like to hear more or connect with any of us, feel free to reach out. We'd be happy to connect you with others and form any sort of partnerships and share the work that we've been doing, or help the work that you've been doing, and really make some connections too. So I wanted to make these contact options available. We also have our social media platforms, and we would love to see you at upcoming community conversations. So with that, I want to be mindful of everyone's time, and I wanted to thank all of the panelists, and the live notes doc will still be open where Marissa can add other information and people can post more questions if they would like to, and our panelists can answer. But wanted to thank our panelists and thank everyone for being here today.